So immune system. Immune system is obviously like protecting you from stuff. You have this kind of warm, moist environment inside your body full of nutritious molecules and stuff. It's be a great place to hang out and multiply if you're some little bacteria or something. And I mean the fact is you do have bacteria that are your friends. We talked about the gut bacteria. So I mean, there are bacteria that are actually work with you, but then there's other things that will multiply and make you sick. They'll take up your resources. They'll create toxins that poison you. Um, there's also multicellular parasites like worms and things like that that you need to protect yourself from. Um, and so one thing that this is protects from what we'd call like pathogens. Pathogen just means like generate suffering. So this would be like bacteria, viruses. Also things like worms or protists, even like giardia. If you've ever worried about getting giardia when you're in the backcountry, that's a little protist. So there's lots of little creatures or things that can get inside that you need to protect yourself from. It's also about protecting you from your own cells. So it's also um, deals with mutated and abnormal self cells. Right? Cells are constantly getting having problems. Like some mutation in the DNA, and they start dividing, um, abnormally. You got to take care of them, or else, if it slides under the radar, you're gonna get cancer. If you have your own cells that start dividing uncontrollably, that's just as bad as having some external cell come in and start wreaking havoc. So your immune system is both dealing with your own cells and sometimes also viruses. If we'll talk a little more about them, viruses kind of get inside your cells and hijack your cells and basically make your cells start making more viruses instead of doing their normal thing. So that's another thing. If you have a cell that's just completely permeated and taken over by a virus, you know, your immune system has cells that come in and just take that, infected cell out of commission. So it's not going to just spread and release viruses to the rest of your body. So again, both dealing with external pathogens, but also dealing with your own cells that have gone rogue. Um, and when we talk about the immune system, there's like two main divisions, and they do interrelate. So we'll talk about kind of how they interrelate as we go on as well. But there's the idea of general defenses, general also called like innate, versus specific or adaptive. And we're going to be looking at both of these, and also, like I said, looking at some of the overlap. General defenses are things that are not targeting some very specific pathogen. These are things that are either very broad, like like your skin. This would be like this would be like locking the door of your house to keep people from coming in, or putting up a fence around so things can't get on your property. Right? It's not about some particular thing. Specific defenses, these are the ones that are going to use antibodies and things like that. These are antigen specific. These are looking for a particular pathogen. This would be like if you had a restraining order against like some stalker or something, where there's this one particular person who is identified as the problem, and that's the one person who is barred from stepping foot on your property, right? That's specific defenses. General defenses is put up a fence and lock the door. That's kind of keeping you safe just in general. So we're going to look at both of these. We're going to start with the general defenses, and then we'll get into the specific defenses. And again, this is going to be, be using antibodies, going to be mediated by your lymphocytes, which we mentioned, but we haven't really mentioned much. We'll talk about, and this is also, each of these things have subdivisions. Specific defenses are going to have cell mediated versus antibody mediated and all that. So we'll go into that in more detail. So let's start here. General or innate defenses. Back. And I hear somebody's not muted because I can hear stuff. Okay. Right. Like almost everything we do in this class, everything is going to be put into divisions and subdivisions and subdivisions of subdivisions. So the general defenses are kind of a subdivision of your overall defenses. General defenses can be subdivided into more of a external versus internal. External is trying to get things from or keep things from actually getting into your body in the first place, right? So this would just include your skin. Your skin, is basically made of these really tough scales made out of this keratin protein that's just making a physical barrier. And this is be your dry skin. The skin that's lining things, are mucous membranes. So we have, mucous membranes, rather than being kind of hard, and they have this sticky stuff that things get trapped in. And not only that, that mucus is usually, and we're going to talk more about, the mucus often has antibodies excreted into it, and also the idea of lysozyme. Actually, I misspelled it. So lysozyme is something that your body makes that actually attacks the cell wall of bacteria. So it basically attacks bacteria. And it is excreted into mucous membranes. It's in your saliva. It's in your tears. So this is another kind of one of these things. If some bacteria lands on a mucous membrane, or there's something in your mouth, or even landing on your eyeball, this lysozyme helps kind of break apart that bacteria. This is, again, we talked about serendipity. Like one of the way they found lysozyme was somebody was examining bacteria under the microscope, and some nasal drippings dropped on the slide, and they realized that it was actually lysing the bacteria. So they realized there's something in your snot basically that breaks apart bacteria. So lysozyme, acidity is part of this external defenses. Most of the pathogenic bacteria don't do so well in a low pH environment. Your skin, the surface of your skin is basically acidic, like from the sebum and everything. Vaginal canal is acidic from having these symbiotic bacteria, these bacteria that make lactic acid. And that lactic acid makes it inhospitable for, like, for instance, Candida albicans, like the yeast, to divide. like if a woman takes antibiotics for some other reason, but that also is going to knock down her kind of indigenous population of these lactic acid producing bacteria, she's much more likely to end up getting a yeast infection, because all of a sudden, like, you know, your friendly bacteria that make it inhospitable to yeast are also getting knocked out by this kind of broad spectrum antibiotic. So overall acidity, and this is also like in your stomach, right? That pH 2 in your stomach is also just taking out bacteria that you might be taking into your body, or that is trapped in that mucus that is getting that's in your respiratory tract, getting swept to the back of your throat that you're swallowing. So acidity is important. Yeah, so I should say, maybe I'll just do that specifically, HCl and stomach. So this is about trying to take things out before they ever cross the boundary and are inside your body. But obviously things do get inside your body, and you have a whole array of different things going on inside your body to deal with things that have made it in. So let's go to the internal. So far this is general defenses, which is this, overall is body defenses, general defenses as opposed to adaptive defenses, external aspects of general defenses. Now we're going to the internal aspects of general defenses. So there's a whole bunch of stuff here, and we'll talk about some in more or less detail. One big thing are phagocytes. Cells that eat stuff that shouldn't be there. In the general defenses, the phagocytes are basically targeting things that just don't look right. Like there are particular carbohydrates that you'd only find like on a bacteria, for instance, that would never be on a one of your own cells. They have, so a lot of these things, they're called toll-like receptors that recognize markers that are on invader cells, right? I talk about, I often say, like, if I'm scanning around the class and I see like a gorn, like there's somebody sitting there at a seat and they're covered in scales with big fangs, and it's like, I know that I don't have to even guess if that is one of the students that belongs in this class or not. Like nobody who belongs in this class sitting at a desk should be covered in scales with big crazy fangs and big glistening eyes or whatever. So some of the general defenses that include phagocytes are just looking for markers that would only show up on an invading cell, that would never be on one of your own cells. The phagocytes also get helped by other aspects of the immune system. There's this thing called opsonization. It means making tasty. There are different things that will opsonize a target. For instance, antibodies do this. Via complement. We're going to talk about this more. This is where I'm saying that there's actually overlap between the different divisions. So one of the things that specific immunity can do is mark things that are specific, but mark them so the phagocytes say, oh, this is something I particularly want to eat. So there's this idea, we're going to talk about complement in more detail in a few moments. Complement gets its name because it complements both sides, specific and general immunity. So etc. So There are things that will help the phagocytes recognize targets. The two main kinds of phagocytes, um, these are both different kinds of white blood cells. So I mentioned we're going to start, when we first talked about blood, I said there's a lot of white blood cells, and we'll be talking about them in more detail. Now is kind of when we're talking about your white blood cells, the leukocytes, in more detail. One of them are called neutrophils. Neutrophils come in and, again, they eat stuff that either doesn't look right or that has been opsonized by these other processes. A lot of the pus, like when you have an infection and there's that white pus, that pus is kind of like the dead and dying neutrophils that have eaten a bunch of stuff and are so filled with kind of toxic things now they're dying themselves. So neutrophils, and they're kind of, when I always, when I think about the immune system, I think about it kind of in like as a military analogy. The neutrophils are more of kind of the infantry, like kind of the expendables that go on the front line and basically kind of die in the trenches there. And again, these things, they actually can be circulating in your bloodstream. And one of the things I mentioned about white blood cells, what blood cells are not confined to the circulatory system. If there is some infection happening, there are these chemical messages that are released, and this idea of chemotaxis, where the white blood cells move towards these chemical signals where they're needed. So the neutrophils will actually like grab onto the side of the blood vessel and ooze out of the blood vessel. They call it diapedesis, and actually get into the tissue spaces where they're needed to fight the infection. The other major kind of phagocyte that is kind of more complicated, I think of these are the ones that are like kind of the officers or something, the macrophages. Macrophages, they're kind of like big amoebas that kind of are moving around. They can, neutrophils, kind of eat, I think, like 25 or so things before they kind of are done. Macrophages live a long time, eat hundreds of things. In addition to eating things, macrophages are also often what we call antigen presenting cells. So, so if I am a macrophage and I'm going around and I am, I can do like, I should. Here's like some bad bacteria that's in my body, and let's say I'm the macrophage, and I go, it's all gone. But then what I do is I actually hold on to a little piece of it and say, oh, this is a very identifying portion of what this thing looked like, and then I'm going to go to talk to like the lymphocytes and other cells in your specific immune system and say, this is an identifying marker for this thing that we want to deal with. And then it's going to start a whole other part of your immune system going, the specific immune responses that we'll talk about later. So macrophages, both they eat things, get things, take care of things, but they also often play a role in actually presenting, identifying markers of what they've eaten, so the specific immunity can then be alerted and come online. Macrophages also release lots of other special signaling molecules. So we're going to talk about fever, They release pyrogens to like reset your body temperature. They release things that actually change your body's, holds on to iron, takes iron out of the system, which makes it harder for bacteria to multiply. There's so many different special communicating molecules that white blood cells use. There's this, you'll often see the word interleukins. Inter means between, leukins, leukocyte. These are just like signaling molecules that white blood cells use to talk to each other and to coordinate their responses. It's going to become even more important when we get to the specific immunity. But there's all sorts of specialized specialized communicating molecules that white blood cells use to coordinate this incredibly complicated response. What else? Let's talk about natural killer cells. NK cells. So these are going to take out like your own cells. If you have your own cell that has been infected by viruses or is kind of mutated in some way that it's no longer functioning properly, there'll often be some message or marker that these can recognize. We're going to also talk about specific immunity. There's going to be killer T cells when we get to your specific immune system. So there's a whole other population of killer cells when we get to the specific immunity, but this is more general. Again, cells that are not functioning properly and are sending out particular things that these natural killers can recognize, so the natural killers know that this cell is infected or malfunctioning. And rather than eat things, what they do is they actually kind of poke holes in their target in order to eliminate the target. So these insert what we call like perforins. And again, the target here is going to be an infected or cancerous cell. And like I've mentioned, cancer is when something slides beneath the radar, but cells are always things wear out, things mutate. And most of the time, your immune system is really good at seeing something's gone wrong and let's get the bad apple out of here. So perforins are basically let's say this is the cell that is not doing so well. So this cell has to get taken out. Your natural killer cell kind of comes in and it puts these little proteins. They're kind of like people talk about them like the staves of a barrel, like the different parts of the different slats and when they all come together, they'll actually kind of make a tunnel that actually makes a hole through the entire membrane, so that the cell is no longer going to be able to moderate its osmolarity, and also these things that release things that kind of come in and help kill it. But so you can kind of think of it metaphorically as it's coming up and stabbing it and poking it full of holes. Although it's not actually using a dagger. It's actually using these little perforins, these little membrane proteins that go in and assemble to create a hole in the membrane. We'll see a few different things that work this way. Like I said, the killer T cells that are part of your specific immunity are also going to do this kind of thing. There, though, they're going to be recognizing cells that are infected with a specific virus rather than this more general like thing. When we get to complement in a few moments, we'll see complement is also working by inserting these membrane proteins that create holes in the target. So natural killer cells, and again, we're going to come back a little bit and we'll meet the killer T cells that are part of specific immunity, but these are general guys. What else do I want to say? Let's talk about complement. Actually, before I talk about complement, I should talk about inflammation because complement deals with inflammation. Actually, if I do inflammation, I should type it in red. Obviously it gets its name inflamed, go, on fire. What are the hallmarks of inflammation?
Redness, heat, pain,
Swelling.
increased function.
So redness, heat, pain. Another big one is swelling. These are kind of like the things that come, and inflammation is something your body is doing intentionally, right? This is not a malfunction. This is actually, we're going to talk about what's actually triggering inflammation. This is triggered, by certain white blood cells, like the mast cells in your connective tissue, or the basophils in your bloodstream release histamine to create inflammation. So there's like mast cells and basophils. This is also triggered by this idea, and again, complement we're going to talk about in a little bit, but these are basically proteins from your plasma. So there's lots of things that will trigger inflammation, and these things come from what's going on. Like this is going to be like kind of, vasodilation. Let me write that better. Part of the redness, and the heat is just dilating blood vessels and letting more blood come into the region. And the vessels get leakier. So having the leakier vessels are letting like the neutrophils get out into the tissue to like fight things, allowing antibodies to get out into the tissue. Vasodilation is letting just more blood come in again to bring nutrients and get rid of wastes that are being created during the fight. So this redness and this heat, are coming from this kind of just extra blood coming in. Having the vessels get leakier, you get what's called exudate. That's basically having more fluid leaking out into the tissue, kind of localized edema, which is causing this swelling. Part of the thing that is part of the exudate, in addition to like antibodies and things like that, you end up having like fibrinogen. Can get out into the tissue. Why is it useful to have fibrinogen like as part of this process going out into the tissue where you're fighting some infection or trying to protect where there's been some injury?
To close the injury if there is an opening?
It's not so much. It's more to kind of wall off, kind of keep the infection, whatever bacteria is multiplying in there, kind of keep it local, kind of create a wall so the battle is contained, rather than just let it keep spreading and getting wherever. So, when we think about an abscess or something, you have the infection is happening, but it's kind of contained in this little area because you wall off. So this walls off the infection. Um, so these exudates are going to have kind of local edema, right? So that's going to cause the swelling. And both, the swelling, which is pushing on kind of local nociceptors, as well as other just signaling molecules released during this, kind of just increase pain in the area. And again, pain in general, pain is a good thing. Pain is like this signal saying, kind of leave this alone and let it heal, right? Don't use this as you would normally use it because it needs to be, it needs a respite so you can have healing going on. Obviously, pain can become problematic if it's, kind of, always happening. Like when people have neuralgia and things where there's just chronic pain and there's no way to try to deal with it, then it can be incredibly debilitating. But in general, the idea of people who have these genetic things where they don't feel pain, it's actually horrible. It's like they just hurt themselves. They can they're cut themselves and don't realize that they're damaging their body, and they can end up really messing themselves up. So in general, having pain is a good thing. It like lets you know something's wrong, lets you know, leave it alone, let it heal. So inflammation, again, it's something that's intentionally being triggered. It's not like things are going wrong. Same thing with fever. Fever is something that is intentional. Fever is not, losing homeostasis. Fever is actually resetting your homeostatic temperature. So fever is actually instigated by these molecules, these signaling molecules released by white blood cells. We call them pyrogens. Pyrogen means to generate fire. So white blood cells release pyrogen, and these talk to the hypothalamus. And they reset your baseline temperature. Remember, we talked about the hypothalamus being your temperature control. Exactly what fever is doing, why it's useful. There's like hypotheses, but nothing, I've never read anything that's super slam dunk, like, oh, obviously that's why fever exists. Again, like I've mentioned earlier in the semester, even things like sweating, it's pretty freaking obvious. Like if you're sweating, it's helping cool you off. It's evaporative cooling. You don't have to think too hard to think like we got a good explanation for why people sweat when they're hot. Why people have a fever when they're sick, that's a little more unclear, like exactly why this happens. But what is clear is that it happens on purpose, and that as long as the fever is not kind of out of control, then you just usually want to just let it play its course because your body is doing it, intentionally. If the fever starts getting above, I forget what it is, like 103 or something, then they start trying to bring it down because you don't actually, you actually can start having the different systems lose homeostatic balance and start getting out of control. But if you have like a low grade fever, you're not supposed to knock it down. You're supposed to let it ride since, your body's doing it. It's got some reason for doing it. So fever.
One question, Professor. Sorry, I thought that fever was mainly to, I don't know if it's true, to kill some bacteria because it's a higher temperature. Maybe some bacteria cannot live on that range of high temperature, or,
Yeah.
or that is not true.
It could be, but then I
Oh,
also think of higher temperature things usually often divide faster. I mean, the high temperature is not that high, you know. So I don't know. I would say if somebody, maybe there's been some stuff more recently where people have a more definitive explanation for why fever is helpful. Um, but the last time I looked, I didn't see anything that just made me feel like, oh, we finally figured this out. Um, so fever. Another important non-specific defense is interferon. So an interferon is actually a collection of molecules, the interferons, but we'll just call it interferon. These are kind of like a viral attack warning. I'm not sure how. I mean, most people know what viruses are. You know, a virus like coronavirus. Coronavirus is this little dealy bopper. You know, everybody sees it with the little things, spikes to make it look like a corona. You know, and it manages to find one of your cells, like in the case of coronavirus, it finds an ACE2 receptor or an ACE2 enzyme on the cell, and it manages to dock, and then it injects basically, genetic material. And then it hijacks, cells, protein synthesis, and DNA replication. And basically it makes, a virus in and of itself cannot reproduce. It's just genetic material with some protein around it, but it's on the verge, like what's alive, what's not alive. They can reproduce by hijacking your own cells. They go and then they take over your own, your cell's ability to replicate genetic material and read off genetic code to make proteins. And then basically your own cells do the dirty work of making a whole bunch of new viruses, which then eventually can get released out to infect more cells. So interferon is something that is released by your cells when they are under attack by a virus. And they basically are telling these other cells, it's like this cell here, it's like, it's too late for me, save yourselves. And so like the interferon is a signal that goes to another cell, and this one's like, whoa, and it's going to basically harden its defenses, kind of slow down protein synthesis, things like that. So it'll make this cell less vulnerable to viral attack. And it's not, it's kind of in the long term, it's stuff that you can't keep up, right? Because cells have to make proteins, they got to do the things they got to do. But in this short term, it's going to take a bunch of measures that make your other cells less vulnerable to viral attack. So that's what interferons are. Interferons are molecules that are released. So cells that are not yet, attacked can make themselves less vulnerable. And these cells that are already riddled with viruses inside, they are going to be taken out by, your killer cells, whether your natural killer cells or when we get to the, killer T cells or your specific immunity. So hopefully before they kind of release all the viral load and continue into spreading it even further. Unfortunately, we could talk more about viruses. For something that are not alive, they are pretty advanced, tricky little buggers. They're fascinating and kind of terrifying. And all over. I mean, most viruses don't hurt you at all. viruses are mostly just affecting, attacking bacteria in the soil or something. Um, there's a whole like food chain. They're not even eating, so I wouldn't even call them part of the food chain, but a whole kind of chain of life, of viruses to bacteria to eukaryotic cells to animals. And it gets really complicated even if you're trying to find like for a plant to be healthy. Often, for a plant to be healthy, it's only going to be healthy if there are specific, bacteria that work along with its roots to help it, get nutrients, or but often those bacteria only work well if there's specific viruses that work with them. So, even them, like you actually have viruses that are kind of symbiotic in your skin to help protect you from bacteria. So viruses aren't necessarily evil. Just like we have symbiotic bacteria, there's evidence that we actually have helpful viruses that protect us from bacteria. So, but bad viruses like, coronavirus, whatever his name, I forget, the one that gives you COVID, you don't want him. So interferons. But yeah, just kind of putting out the web of life is way more complicated than just us and them. There's a lot of things that are either actually you work with in some positive ways, or they're there but they're irrelevant. So it's not like everything's gonna kill you or something or make you sick. All right. The last part of the specific, the general defenses is going to be complement. So this is again this is kind of general defenses, but is also going to be recruited by specific. So I think that's why it gets the name complement, because it complements many different aspects of your immune system, but it's kind of a stupid name because it's so generic. Because it's actually something. Complement is a bunch of, this is like 20 plasma proteins. That are activated in a cascade. Kind of like when we had blood clotting, there was a whole, cascade of blood proteins that ultimately resulted in the fibrin clot. This is going to be a whole bunch of plasma proteins that activate. And again, they can be triggered. Like I said, they can actually be triggered by the specific immune system, by antibodies. This can be triggered just because something doesn't look right. Something has the wrong marker on it. So a variety of things can trigger this complement cascade. If complement is triggered, it does a few things. One is it intensifies inflammation. So all those things that we were talking about that are part of inflammation, like making the vessels leakier and dilated, bringing more antibodies and cells into the region, that's all happening. Opsonizes the target. So again, this is that thing basically kind of, gives handles for the phagocytes. So opsonization is basically like putting these big signs all over, like, eat me, eat me, eat me. So all of a sudden, this whatever is getting whatever is getting attacked by complement, all of a sudden has these little handles all over it that the phagocytes recognize and go in and, with extreme prejudice, go and try to eat this thing. And then finally, it creates this what's called a membrane attack complex. And this is basically makes holes in the target membrane. So there's three big things that are happening with complement here. This is a big one. So complement has been activated. Let's say you are the target of complement. All of a sudden, holes are opening up in you. It's like, oh, bad scene. And now you're getting all these like markers on you, and the phagocytes are perking up and looking and coming in to eat you. And again, the whole area, the inflammation's happening, so more and more antibodies and cells and things are coming out of the bloodstream to flood the area. So there's all these things happening in parallel that are all working to, basically neutralize the target. And again, when we get to the specific immune system and talk about antibodies, a big thing that antibodies do, in our blood lab, we saw antibodies can do agglutination, right? When antibodies bind their target, they can cause agglutination. The cells all kind of clump together. That's a useful thing, for our testing to see blood type. You can see agglutination easy. But another thing that antibodies are doing when they bind a target, specifically when there's, it's in your blood, is it's going to, what they call fixed complement, is going to start this whole complement cascade. So when the antibody binds the target, it's going to start this whole thing that's going to create all these things as well. So it's not just about agglutination. When an antibody binds the target, it's starting this complement, which is going to cause holes in the target, make the target tasty for phagocytes, intensify inflammation in the region. So just kind of putting that out there. That's kind of spoiler alert. When we get to how do antibodies do their thing, a big part of it is by what we call fixing complement, kind of activating this complement cascade. Alrighty. So, are there any questions about general immunity? So again, or innate. Again, these are all things that are working kind of all the time and are not designed to find a particular, it's not looking for coronavirus or not looking for, poliovirus or this or that. It's just looking for things that don't look right in general, or cells that are sending out messages that I'm hurting in general. Or some of them are even just about barriers, right? Kind of mucus and skin and things like that. So what we're going to do now is shift gears, and we're going to talk about specific or adaptive immunity. So this is going to be the immune system branch that does deal with a very specific kind of target. So let's do that. So again, I've mentioned antigens. I'm going to mention antigen again because none of this makes sense without the idea of antigen. So Here's some nasty little beastie that is inside you that might make you sick. And it has a really specific kind of look, right? It's got membranes, and those membranes have different carbohydrate groups hanging off of them, different cell markers. And those markers are things that can be recognized by your immune system. So this whole thing, this thing that is going to, this is going to be like my antigen. And it basically means antibody generating. It's basically anything that has kind of specific shapes that you can recognize. The specific kind of regions that you can get off of it, these are kind of called antigenic determinants.
Is an antigen the whole cell?
It's like the whole target, right? So you can actually have more than one you can have different antibodies that recognize the same target, the same antigen. Like if you ever talk about, you hear like polyclonal antibodies versus monoclonal antibodies. Polyclonal antibodies are where you have a whole array of antibodies that all might recognize slightly different shapes, but are all recognizing the same overall target. Whereas monoclonal antibodies are basically all off the same template and are all looking at the exact same antigenic determinant. And again, given if we had more time, I would spend more time talking about that, but it's something you'll look at later. So this idea of antibody is going to be important. Again, it's something that is a particular thing and that has particular regions that can be recognized. So when we talk about specific or adaptive immunity, there are three main things that characterize specific or adaptive immunity. And the first one is it's antigen specific. So if you are creating a response against coronavirus, whatever, I forget what the version is, this SARS-2, blah, 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 whatever. What is the name of this current one?
SARS-CoV-2, like SARS-CoV-2.
So SARS-CoV-2, if you're creating if you've been infected by it, it's going to be specific to that virus. Or what the vaccine is doing is creating a specific immune reaction against that specific antigen. So antigen specific. Another thing is it's systemic, meaning it doesn't matter where this antigen initially was encountered in your body. You're going to create these cells and antibodies that are everywhere. So wherever this antigen is encountered, it will be recognized and neutralized, right? This other stuff we've been talking about, inflammation, that's happening right where the infection is happening. It's not going everywhere. So it doesn't matter where your body first encountered the antigen. Once you have initiated this specific immune response, wherever that particular antigen is found in the body, it will be recognized and attacked. And then the last part of this is, again, it's why why we can have vaccines, is it has memory. So memory meaning that after you have done fighting this thing, you actually squirrel away a template of what this antigen looked like. we're going to talk about them specifically. We're going to have memory T cells and memory B cells. That these are memory T lymphocytes, memory B lymphocytes that are just kind of held back in storage. And if you ever see the same antigen again, your body knows what it looks like, already has the template to work from, and can ramp up a defense much faster and stronger than if you had encountered it for the very first time. So in fact, it's worth just kind of talking about this more specifically now. Yeah, like 1, 2, 3. You know, my y-axis is this very vague immune response. So let's say on day 1 here, you encounter the pathogen. We're going to talk about, there's a bunch of steps that have to happen. You know, your white blood cells have to eat it and present the antigen to the lymphocytes. And the lymphocytes have to start kind of doing what we call it clonal selection and multiplying and creating all, you know, so this is like first encounter. The first time you see something, it takes a while for the immune response to really get going. And in the meantime, this pathogen is multiplying and you're getting sick. And eventually your immune response is getting stronger and you knock it down and you get better. But the fact is you got sick in the process. If you now encounter that same pathogen another time, so the second encounter, again, this time you have memory because you've built up these memory cells. So the second time you see it, it's much stronger and faster. And basically what's happening is this thing comes in, and before it has a chance to really get a foothold and make you sick, it's been knocked out. So this is when we talk about immunity. Immunity means you have these memory cells that are lying in wait, and when that particular pathogen is encountered in your body, you don't have to wait for this kind of long process to, you know, ramp up the production of all the antibodies and stuff. You just have this fast, strong response that knocks it down so you never even realized you were going to get sick. You know, that's what, you know, normal immunity is. Vaccines work by trying to simulate the first encounter without ever making you sick in the first place. So for vaccines, you know, you basically, you have this thing, like let's say this is a thing that would have made you sick. You can use like killed virus. You can use viruses that look like the real thing, but actually are not pathogenic, aren't going to actually make you sick. So your body sees this thing, has a response, and you might feel a little malaise. Part of the those signaling molecules, those things released by white blood cells, in addition to doing everything else, they make you just kind of lethargic and tired and want to just lie around. You know, it's another part of healing is like not running around. So lying, and so you might still feel kind of malaise, but you're not going to get sick in the way that if this thing was multiplying. So you can have like killed viruses like this. You can have just attenuated viruses, like looks like it is, but it might actually multiply a little bit, but really not bad at all. So it never makes you really sick, and your body has a strong immune response. So if the real stuff ever shows up, you will be able to take it out. There are also things that are just based on parts and pieces. Like you put something in that is just a piece of what this thing looks like. And, it's a tricky thing because it's got to have enough of a presence that your immune system recognizes it as something to make a immune response against. So kind of coming up with a vaccine is tricky because you have to have something that is going to trigger the immune system to make memory cells that are going to recognize the active, really mean version of the thing, but do it in a way that's not going to actually make you sick, and also do it in a way that's not going to accidentally create an immune response that is going to be attacking things that you are going to regret. There's all sorts of autoimmune diseases that happen if you have the immune response attacking things that shouldn't be a problem. I'll talk about that, but basically, and even allergies are part. Maybe I'll talk about it now, like kind of problems with the immune system. If you can have autoimmune. This is where you are specific immunity. Normally it should be about some pathogen, but if specific immunity is attacking your own tissue, you're in trouble. This is what happens in rheumatoid arthritis. You are attacking your synovial membrane in your joints. This is what happens in lupus. You're attacking just your own tissues and just getting overall inflammation all over your body and your organs. This is happening in multiple sclerosis, where you're attacking the myelin sheaths on your neurons and stopping the transmission of impulses along your central nervous system axons. So, all of this can be horrible. And you want to be super careful that you don't accidentally create some situation where you have created some population of lymphocytes that are actually targeting your own tissues accidentally. There's some spillover, some crossover between what you thought was your target, and it also looks enough like something that you didn't mean to target, but now it's, so when you're designing vaccines, you got to be super careful that the immune system is kind of this real balancing act. You want it to be strong enough that it attacks and takes out the things that are dangerous, but if it's overactive, you are going to start actually causing problems and attacking things that you don't want to attack. Like allergies are another. Allergies are this over response to something that really isn't a problem, like pollen or cat dander or something, is not going to hurt you or kill you, but all of a sudden you've got all these mast cells degranulating and releasing histamine, and you've got horrible inflammation, and your eyes are red and your nose is running, and so this is immune response kind of going off in a south direction. Transplant rejection is another thing that is problems. Like sometimes you want some other tissue, like you got a heart transplant or a liver transplant, and your immune system says this doesn't look right, so you're going to attack it. If you do get a graft, or if you do get a organ transplant, you're basically going to have to suppress your immune system for the rest of your life, which makes your life way trickier. You're still alive because you've got like somebody else's liver, but now you are on immunosuppressants, so you have to be careful about ever going to a party again because you might catch something that's going to kill you. So again, immune system is something that is really complicated, and things do go wrong with it, obviously, because autoimmune diseases are not that uncommon. So back to here. So this idea of immunity, where you're using some vaccine or whether it's just because you got sick from it, this is what we call active immunity. Active immunity means you actually encountered this thing either through an actual real encounter or through a vaccine, which was kind of less pathogenic encounter. And in both cases here, in active immunity, you've created memory cells. And so this is going to be long lasting. Sometimes you need boosters and things after a while because the memory starts fading. But in general, active immunity, you have memory cells and it's going to last a while. The other form of, we should mention, does anybody know why they're called vaccines?
Because the first vaccine was on a cow for the smallpox, so vacca, and then vaccines.
Exactly, it was from vacca. Basically they found that cowpox, which was a less kind of virulent disease than smallpox, would protect like the milkmaids from actually getting smallpox. So the first vaccines were like using this less pathogenic virus to this less pathogenic pathogen to protect someone from getting the worst version. But yeah, so it's always kind of cracks me up that vaccine comes from vacca. So, so passive immunity. So passive immunity is something very different. This is where you get antibodies from outside. And you basically have temporary protection. As long as the antibodies are in your body, you are going to be protected from whatever those antibodies recognize. But, after a while, they're going to clear out of your system, and there is no memory here. Because you got some other antibodies, you didn't make these antibodies yourself. It wasn't the normal process where you make antibodies and make memory cells. So this is giving you temporary protection. Like examples where this happens is just in nursing. When you, in the mother's milk, you can actually get antibodies from the mom that will give the baby temporary protection from whatever kind of pathogens are in the environment that the mom has antibodies against. So while the baby's immune system is kind of coming online, the baby is actually going to get temporary protection from the mom's antibodies that they're getting. You can also just get shots of this to protect you if you're in a dangerous situation. Like, antibodies are also called gamma globulins, also called immunoglobulins. So, like when I was in grad school, we had a big dive trip down to Baja, and I got hepatitis A, which was horrible and super, super, super sick. And all of my roommates had to go in and get gamma globulin shots against that virus. So that would keep them safe. They never got sick. I got super sick. But, at this point, I'm never going to get Hep A again because I actually went through the whole process of getting really sick, but then making memory cells. Whereas, the roommates were protected in the moment. They didn't get sick when they had this kind of infectious person in the house with them, but they never made memory cells. So at this point, they still have to be careful about catching this disease. So that's this idea. Passive immunity, it's used. It's used a lot to provide temporary instant protection. But, the downside is, you don't have long-term protection. So, any questions about that? So you should definitely understand the difference between passive and active immunity and what a vaccine is, because especially now, vaccines are so, it's, and part of the reason, the fact that people are actually managing to come up with a vaccine this fast is amazing. And the fact that they're not just releasing it like let's just release it, that they're really thinking like, is it really safe? That's something you really do want to pay attention to, because if they work well, they're amazing. But you don't, you have to be very careful when you're working with the immune system. So what I'm going to do now is, we're going to talk about the two specific branches of the immune system. And we're going to, so we have a little bit more to cover. And actually, maybe before we do that, I'm going to do one last more generic thing, and then we'll take a little break, and then we'll talk about the two divisions and then kind of tie up the immune system. So let's talk about MHC, Major Histocompatibility Complex. So this is a membrane protein. So here's your cell. Whoops. So we're going to talk, there's actually MHC1 and MHC2. I'm going to start talking about MHC1. MHC1 is on almost all cells. Whoops. I think it's not on your red blood cells, but it's on almost all cells. On almost all your cells. MHC1. So it's got two things going on. One is it is self-antigen. Self-marker. This is the thing that your immune system uses to, it's kind of like the barcode saying that cell belongs to me. If you have a cell in your body that does not have that particular version of MHC, then your body says this doesn't belong here. That's why you have transplant rejections, right? Even if I wanted to give you my liver or my kidney or something, your body would say these cells don't have the right MHC marker, and I'm going to attack it and try to kill it. If you are going to do a transplant, you try to get someone that's genetically as close as possible to minimize that response. And even then, you're still on immunosuppressive drugs for the rest of your life, trying to keep your body from rejecting the transplant. So part of MHC is it is the marker that tells your immune system that this cell is part of my body. Right? Histo is just tissue. This is your tissue, your cells. The other thing that it does that is kind of cool is it has this little slot where it puts a little peptide. So this is like, how many? I think 8. Let me look it up to make sure I 8 to 9 amino acid peptide fragment. So endogenous meaning something that was just found inside the cell as this MHC was being produced, right? So MHC, it's a membrane protein, right? So your cell is at the ribosome and it's assembling this thing and probably going through Golgi apparatus, like modifying it and doing all this stuff to make this protein. In that process, it randomly snips some little piece of peptide floating around in the cell. And so this is like some random peptide fragment from inside, and it's displaying it on the outside here. So it's basically saying, here's a little sample of some random sample of what's found inside of me. It would be like you had a little chunk of your heart, or a little chunk of your liver, or a little chunk of your spleen or something kind of on display, saying, oh, this is a little example of what you find if you went beneath the skin and looked inside of me. Why is this a useful thing?
Like if you have a virus, it would alter what that would look like.
Right. So if you are, let's say you are actually under attack by some virus, and actually it's not all okay inside of you right now. These MHCs are getting produced and taking some random fragment from inside, and the thing that's showing up out here is, so what's going to happen is your killer T cells, part of, the killer cells that are part of your specific immunity, my little killer T cell here, he's coming in and he's got like the warrant. He's like, I'm looking for anything that looks like this. This is the invader, part of my specific immunity. I know I need to like find this thing. And he's like, oh my God, this thing is infected with this. So it knows, like, again, it's not going to actually use a dagger. It's going to use the perforins and stuff, but it knows that this cell is riddled with viruses and is going to have to take it out. So this MHC1 allows your immune system, your specific immune system, to actually get a sample of what's inside the cell and see if there's something in there that shouldn't, that is being, part of what the specific immunity is on patrol for. So does that make sense? So like a killer T cell would say, like, if he walked by and it just was some random thing, just leave it alone. It all looks good. But if he sees the specific thing that he's trying to, the piece, the specific marker from the pathogen that, then he knows, like, oh, this is bad. Just very briefly, I'll say MHC2, and then we'll take our little break. And the basic idea here is this presents, presents an exogenous peptide. And this is kind of what I was just talking about. I'll just draw more clearly. here is your, like, let's say this is a macrophage. And here is the little beastie. Rawr. And this is going to be a phagocyte. So this is going to come in and come around and eat this thing. So the this macrophage ate this thing and is kind of dismantling it, but it's going to keep a little fragment of it. And I didn't mean to be that fat. It's going to keep a little fragment of it. So, whatever this thing was is just getting whatever. But now we have this MHC2. And it's going to present a little piece of what it ate. That's what I'm saying, presents an exogenous peptide fragment. It's saying, here's a little sample of this thing that was invading that I ate. And after break, what we're going to see is this cell then is going to go and present this to the specific immune system and say, this is what this invader looks like. Please set up a specific immune response against this particular antigen. So that's what MHC2 is. MHC2 is this mechanism only found in specific white blood cells whose job it is to activate specific immunity. And it basically presents some little piece of something that the cell ate in order to activate specific immune responses. As opposed to, MHC1 here, which is basically on all your cells, except for like a few. And that's more about general self-marker as well as presenting some little fragment, random internal peptide to let the immune system know whether or not everything's okay inside the cell. Okay, so let's take a break. We'll come back at 10 and we'll just tie up with a little bit more detail about what exactly are the two divisions of specific immunity, so you kind of have a more complete sense of that. All right, so we are gonna kind of finish up by getting a little deeper into the two divisions of the specific immunity. So again, back. Specific, aka adaptive immunity. And like I mentioned, what are the three hallmarks of specific immunity?
Memory.
It always has memory. What else does it?
Works fast in the second exposure with the antigen. It has fast response.
So that's more about memory, right? That's an
Okay.
Antigen specific and systemic.
It's antigen specific and it's systemic. It doesn't matter where you first encountered the antigen, you'll have a response across, in the entire body. So just kind of keep in mind that these are more general properties of all branches of specific immunity. What we're going to do now is kind of drill down and look at the two sub-branches of specific immunity. So let's do that now. It's going to be antibody mediated also called humoral. Humors are kind of an old word just for the fluids of the body. So humoral, it's like, so antibodies are these little proteins that are being just kind of, put out into the humors of the body, so to speak. That's why it gets that kind of name. The other one is called cell mediated. And so now we're going to talk about how these two different sub-branches are kind of unique from each other. Antibody mediated, just from its name, you can kind of guess, kind of uses antibodies, to, to target, or maybe, how should I say this? Uses antibodies to attack target. And again, antibodies are, they're plasma proteins. We'll talk a little more about them, but they're basically plasma proteins that are being made by cells. Cell-mediated, cells themselves are used to attack the target. This is where we're going to have like the killer T's and stuff. Killer T cells. Other differences is what subpopulation of lymphocytes is used for these two different sides. Antibody mediated uses what are called B lymphocytes. B is because they mature in the bone marrow. That's why they get the name B. B lymphocytes mature in the bone marrow. Sometimes they're just called like B cells. The cell-mediated is done by another population of lymphocytes that mature in the thymus gland. This kind of gland over your heart here. These are called T lymphocytes. T cells. So that's kind of the big difference between them, is what subpopulation of lymphocytes they use, and then also kind of how they attack their target. Antibody mediated uses antibodies, cell mediated uses cells. That should make it easy to remember there. So what I'm going to do now is get a little more into detail. Let me look at the time. I'll talk briefly about how this whole system works. So you might think that the system works like, let's say there's, again, this pathogen, let's just bring in the same dude. So you might think, you capture this, you've got a piece of it, it's going to be efficient to try to manufacture something that actually fits that, but that's not how it works. Instead, your body just makes just tens of thousands or more of all these random receptors. So like, you'll have B cells, and they all have these receptors that have these randomly generated shapes that they could recognize and bind to. Oops. So these are all just kind of floating around and just waiting for their moment to shine, if maybe they're gonna be the chosen one or not. But these are just all generated randomly and are just floating around, not doing much of anything. So then what happens is some antigen presenting cells, some macrophage comes in and eats this thing, and then comes in with its little MHC presenting a little piece of, maybe a little piece of the tail here. So here's my antigen presenting cell. Here's my little MHC2, which is presenting a little piece of the thing it just ate, right? This thing just gobbled this. And now it's like, let's take a look here. Does this match that? Nope. Would this fit in that receptor? Nope. This one, it's like bingo. You know, this thing, the receptor and the antigen match. So what happens now is we know that this B cell had the template to make a receptor that matches the antigen, the thing that we're trying to fight. So what happens is this B cell becomes the template for a whole clone army. This would be like Jango Fett in in Star Wars, being the template for the whole Republic, clone army, right? It's one thing that is optimal to fight the fight. It recognizes the thing we're trying to fight. So this is going to undergo what's called clonal selection. We're going to take this one B cell that had the receptor that has just the right shape, and we're going to make a whole bunch of cells that are all programmed to make antibodies that have that exact same shape. And so then I kind of go back over to here. There's going to be this idea of clonal selection. This is assuming that this match has occurred. And I should say that there's a lot of checks and balances and fail-safes before you actually start clonal selection. This match here has to be checked and double-checked and triple-checked, and make sure that it's not going to actually attack your own body, blah, blah, blah. There's all these handshaking, these cytokines that are sending... This is sending a message back, and this is sending it back, and there's actually going to be helper T cells double-checking both sides. So we're not going to get into it other than it's super complicated, and we're still figuring out the details of it. Things like most of the stuff we're talking about, we have a pretty good sense of. The details, the really kind of more subtle details of what's going on here in the immune system, I swear every week our understanding keeps changing and becoming more kind of deepened. So I'll just put it out there that it's really complicated, and there's a lot of checks and balances. And clonal selection starts, and we're going to make two populations of cells that have that same template, that recognize that antigen. There are going to be what are called plasma cells. And these make antibodies. Like about 2,000 antibodies per second. So they are just going to, again, we've seen antibodies in the blood lab, right? They're little plasma proteins that have these variable regions that will recognize the antigen. And so these are spewing out all these antibodies. These are in your lymph nodes. Like when you have an infection and your glands are swollen, it's because you have all of these B lymphocyte plasma cells that are multiplying and making all these plasma cells, and all these plasma cells are just spitting out all these antibodies to flood your system. So part of this process is flooding your system with antibodies that recognize the target that's initiated this whole clonal selection. And then the other thing that you'll make for the future are the memory cells. And the memory cells are going to wait in the wings, and they're going to provide immunity. We talked about that. They're not really that interesting in the moment, but they're what's going to keep you from getting sick from the same thing next time, because you now have the template squirreled away in the system that's set for rapid and strong response. So let's talk a little bit more about this. The plasma cells making antibodies and why that's useful for protecting you. So it's a protein. It's got these variable regions, that binds the target. And you have them in your bloodstream, you have them in your tissues. Again, they can get excreted into your secretions like your saliva and your mucus and things like that. So, but the basic idea, they're proteins being made by these plasma cells that have whatever shape, whatever antigen triggered that whole clonal selection. Each of these antibodies has this thing that is going to bind that antigen. And so then you might ask, what's the big deal? How does this destroy the target? Like, let's say I am, the nasty little dude here, and an antibody, it's like, he's like, oh, I'm so scared. This little antibody just stuck on my tail. Why is that going to help, destroy this thing? There's like three main things that, and again, two of them we've talked about already. One, antibody binding the target. One is, fixes, kind of activates complement. And I talked about complement, that cascade of events that will opsonize, will poke holes with the membrane attack complex, intensifies inflammation. So this little critter here is like, he's like, oh, do I care? This thing is on me. It's like all of a sudden he really cares, because membrane attack complex, we got holes coming in. And opsonization, all of a sudden he's tasty for the phagocytes coming in to go and eat them up. So having this complement activated is really going to help take out the target here. Another thing that the antibody binding does is the thing we've already seen in the blood, agglutination. Basically a bunch of these guys get all kind of stuck together, and makes it easier for the phagocytes to come in and just grab them all. So all of a sudden they're all stuck to each other. And then the last part is what was called neutralization. Kind of block critical sites. So for instance, coronavirus, it has a thing that recognizes ACE2 and binds and grabs on and gets to inject into the cell. There's a particular part that binds and recognizes. If that was gummed up with antibodies, the thing couldn't do its job. Think about if you're some axe murderer coming in to go murder things, and all of a sudden, like a sticky wicket landed on you. You're just like, whatever, a sticky wicket landed on me. But if all of a sudden they're like on your eyes, I can't see. They're all over your hands. I can't actually use my hands to pick up my axe to like kill anybody, right? So neutralization is this idea that these little antibodies, these little sticky wickets, start gumming things up that are critical for the functioning of the target. In fact, if you ever get bit by a rattlesnake and they give you rattlesnake antivenom, it's actually just antibodies that bind critical parts on the venom molecule and keep it from doing its venomous job. So neutralization is just this idea that you're kind of gumming up important sites on the target and inactivating it. So does that make sense? Very briefly, I will discuss there's actually 5 main kinds of antibodies. And like I mentioned, antibody is also called immunoglobulin. So we talk about IG. Let me see. So let's do IgM. In fact, I'll put them in the name of the mnemonic. The mnemonic is MADGE, right? It's like, I can't believe I'm soaking my fingers here in Palmolive dishwashing detergent. Yep, whatever. Or she's like serving you coffee at some truck stop or something. IgE and IgD, IgA, IgE, MADGE. That is the mnemonic people use to remember the different kinds of antibodies. It's worth knowing at least a couple of them. IgM, this is the one that is released on first exposure. When you have a second response, like we talked about during immunity, that is coming from the memory cells, that is going to be IgG. This is useful, right? Somebody is coming in with herpes, and you want to know, did they just catch this? In which case you're really going to have to do some contact tracing and whatever. Or is this a recurrence? They didn't actually realize when they first caught it, and herpes is something that has comes back every once in a while. Is this just a relapse, or is this an initial infection? They can check what kind of antibodies are showing up. If it's IgM, then you know that they just caught it, because this is their first time they're ever responding to it. If it's IgG, actually they caught it a while ago, and this is just a recurrence. Their immune system is responding to something it's already seen and responded to. I can take credit. I made this up. The way I remember is like in Spanish, like primero with an M versus like segundo would be second with a G. So that's how I remember this one. Other ones, IgA, this one is secretory. This is antibodies that end up in secretions, like in mucus or in mother's milk or things like that. So so you actually have antibodies also as part of this, external protection to some degree by kind of lying there in wait for things that land on your goo. IgE, this one is often implicated in allergies. I can't tell you much more detail. I just know that IgE is often problematic in that it gets involved with when you have kind of overactive response where you end up getting all itchy because of some cat. IgD is more specialized. It's only found on special lymphocytes, so don't even worry about it. But just to give you a sense, and there's more we can talk about them and how they look and how if they're dimers or pentamers. Like IgM is actually 5 tail to tail, looking more like a big snowflake or something. But they all do basically the same stuff. So that is antibody mediated. Antibody mediated is going to be clonal. There's going to be clonal selection when you actually, activate the correct B cell, make the plasma cells, which make all the antibodies, which do everything we've just talked about, as well as memory cells. On the T lymphocytes, it's kind of the same basic idea. There's also clonal selection. Instead of making plasma cells, because there's no antibodies, it's cells that are going to be used, we're going to make killer T cells. And they're also called cytotoxic, or CD8. These are basically like the natural killers, but instead of poking holes in a target just because they don't look right, they're poking holes in something that is displaying the particular antigen that they are programmed to recognize. So they are supposed to recognize some particular virus, and they see some cell displaying that particular virus, they're going to take them out. So killer T cells poke holes in their target, and usually it's like some cell that's been riddled with some virus that it's recognizing. Again, they also are called cytotoxic because they kill cells. CD8, the name CD8 comes from, I think, cell differentiation. It's a cell marker that you find on them called CD8. Another thing that are made by these guys, in fact, let me go into another. This is cell mediated. This is going to be clonal selection. So killer T, also called cytotoxic, or CD8. Another cell that's made, obviously, are going to be the memory cells, because this is also going to have memory, just like the antibody mediated. And then another kind of cell that is critical here are the helper T cells. These are also called CD4. These are actually necessary to activate both branches of specific immunity. These are the ones that are attacked by HIV. The human immunodeficiency virus. The reason why, that is such a horrible disease is there's two things about it. One is the cell that is being taken out by this virus is the very cell that is at the core of activating your specific immunity. And then what's worse about it is the HIV quickly changes its outer coating, quickly mutates. Right, so if you have a cold this year, you might get a cold next year, Why is that? How is it that you get a cold this year and you supposedly have memory and yet you get a cold next year?
It mutates.
It mutates. So that rhinovirus looks a little different than it did last year, and now your memory cells don't recognize it. But with HIV, it happens so fast that it mutates before the infection is done. So you're getting sick, and then this thing is mutating really quickly, and all of a sudden your immune system doesn't recognize it anymore and has to launch a brand new immune system response against this new looking thing. And then it slides under the radar again as it mutates, and all the while it's destroying your basic immune system ability to fight anything. So, if this goes unchecked, that's where you end up with AIDS, the acquired immunodeficiency system, where not only are you getting this thing taking over, but you can't fight anything. You're getting horrible infections from things that normally are just random little things floating in your environment. So, yeah, no, that's this one's particularly bad. Flu, getting the flu shot, what they're doing is they are looking at what does this virus look like where it, on the other side of the world right now where it's currently infecting a bunch of people. And let's quickly ramp up production of some vaccine that will protect you from what's likely to make landfall in North America 6 months from now. So helper T cells, just kind of putting that out there. Helper T cells are necessary for activating both branches of specific immunity. And when you think about AIDS, they're the ones that are getting affected by HIV, which makes HIV particularly nasty because it's dismantling your immune system. Dooby-dooby-dooby-doo. There are other cells. There are suppressor T cells, this or that. Don't worry about it. The main T cells that, be aware that the immune system is really complicated. And some of the stuff is kind of hard to even figure out. Like sometimes repeated exposures to things, you get hypersensitive and you get even more sensitive to things. Sometimes repeated exposure to things, you become less sensitive to things and you don't respond to them. And what causes one path versus the other path, it's hard to know. Or it's really, the immune system is still something that is very mysterious. Anaphylaxis, I should mention anaphylaxis just super briefly. Anaphylactic shock. This is kind of like an overall inflammation across your entire body. Normally like an allergy is like, oh, my eye got red and itchy, or my nose got red and itchy, or my skin's getting hives or something. If this is kind of just this kind of systemic going everywhere and you have like systemic vasodilation, your blood pressure is just going to plummet. You're not going to be able to keep blood flowing to all your tissues. That's anaphylactic shock. Right? Shock is when you can't supply blood to your tissues. What do they usually give you if you're anaphylactic shock?
Adrenaline?
You get like a shot of adrenaline. Your little EpiPen that you jam into your leg is basically putting adrenaline into your body and basically vasoconstriction and speeding up the heart and trying to bring your blood pressure back up so you start getting the blood flowing again and you're perfusing your tissues again and all that. So so anaphylaxis is kind of like an allergy that is like your body rather than just like some local thing. So yeah, and obviously you can die from this because you're in shock. So So he's like trying to say he was just playing a role. He's actually part of our, helping us teach the immune system. He's like, I'm not killing anybody. I'm just wearing my little suit. He's waving goodbye and saying, we are done with the immune system here.